Hello dear viewers, a warm welcome to all. In this module, we are discussing metal nanoparticles, their synthesis properties, characterization methods and applications. Also some discussion on metal nanoclusters. First of all, metal nanoparticles. They are represented as MNPs. Metal nanoparticles are nano-level entities made up of pure metals. The pure metals include gold, platinum, silver, titanium, zinc, cerium, iron and thallium. Metallic nanoparticles are considered flexible nanostructures because of their ability to control their composition, shape, size, structure, encapsulation possibilities, assembly and their optical properties during the course of the synthesis. The existence of metal nanoparticles in solution was first recognized by Michael Faraday in 1857. We all familiar with the experiment Faraday's divided metal. Faraday's divided metal comprised of gold nanoparticle. Main characteristic features of metal nanoparticles. The metal nanoparticles are associated with very large surface area to volume ratio when compared with that of bulk counterpart. They have large surface energies. The transition between molecular structure and metallic states providing specific electronic structure, local density of state. They are associated with plasmon excitation. In metal nanoparticles, there is a quantum mechanical feature called quantum confinement effect. They have short range ordering as far as their crystal structure is concerned. A large number of low coordination sites such as corner and edges having a large number of dangling bonds and consequently specific and chemical properties and the ability to store excess electrons. These are the characteristic features of metal nanoparticles. Now, we can have several methods for the preparation of metal nanoparticles. Let us have an outlook on the chemical and physical methods employed for the preparation of metal nanoparticles. Regarding the chemical methods, there is chemical reduction of metal salt is there, microemulsion method is possible, thermal decomposition of metal salts and electrochemical synthesis. These all belongs to the category of chemical methods. Whenever we are outlooking the physical methods for the metal nanoparticle preparation, there will be exploding wire technique, plasma method, chemical vapor deposition technique, microwave irradiation, pulsed laser ablation, sonochemical reduction and gamma radiation techniques are possible. Features of a good synthetic procedure. The synthetic procedure adopted may be either physical or chemical route. Whatever may be the route, a good synthetic procedure must have the following qualities. The procedure adopted should be reproducible. It may control the shape of the particles. The method should be easy and cheap. It use less toxic precursors. Use the least number of reagents. Use a reaction temperature close to room temperature. That is, high temperature or low temperature is not a favorable condition. With a few synthetic steps as possible, minimizing the quantities generated by products and waste. Stabilizing of metal nanoparticle. The metal nanoparticle synthesized by either physical or chemical route must be stabilized by using suitable stabilizing agents. Different stabilizing methods are by placing them in an inert environment like an inorganic matrix or by using a polymer. Stabilization can also be achieved by adding surface protecting agents like inorganic capping materials like citric acid or citrate coating, organic ligands, etc. Characterization of metal nanoparticles. After the synthesis, we have to characterize the obtained metal nanoparticles by different suitable methods. Common methods employed for the characterization of metal nanoparticles are shown in this chart. For particle size characterization, we can use gas adsorption, selectivity poisoning, electron microscopic procedure, X-ray diffraction studies and magnetic measurement. Surface area can also be measured by these techniques. Surface composition, surface complexes, etc. can be measured by techniques like AES, SIMS, UPS, Mossberg spectroscopy, work function EPMA, extended X-ray absorption fine structure, that is EXAFS. Then further spectroscopic characterization like IR, UV, ESR, NMR and different other characterization methods are used for the complete identification of the properties of the metal nanoparticles under our consideration. Now we are exclusively focusing gold nanoparticles. Gold nanoparticles or colloidal gold have been utilized for centuries by artists due to their vibrant colors produced by their interaction with visible light. Unique optoelectronic properties of gold nanoparticles have been researched and utilized in high technology applications such as organic photovoltaics, sensory devices and sensory probes, as therapeutic agents for drug delivery purpose, electronic conductors and also in catalysis. Regarding the synthesis of gold nanoparticles, several methods are possible. Generally, gold nanoparticles are produced by reduction of HAUCl4, that is nothing but chloroauric acid. After dissolving chloroauric acid in the solution, it is rapidly treated with suitable reducing agent. It should be noted that in HAUCl4, gold is present in +3 oxidation state. The treatment with suitable reducing agent causes reduction of Au3+ to Au+. Then a disproportionation reaction occurs in the medium whereby 3 Au+ ions give rise to Au3+ and 2 Au0 atoms. Disproportionation, as we all aware, is the oxidation and reduction happens by the same substrate. The Au0 atoms act as center of nucleation around which further Au+ ions get reduced. To prevent the particles, metal nanoparticles of gold from agglomeration, some capping agent is used. A popular method, a well-established method for the preparation of gold nanoparticle is the Characterization method. It is the simplest method available. In general, it is used to produce modestly spherical shaped gold nanoparticles in water which is of having size around 10 to 20 nanometer. It involves the reaction of small amounts of hot chloroauric acid HAUCl4 with the small amounts of trisodium citrate solution. The colloidal gold will form because the citrate ion acts as both reducing agent as well as capping agent. In certain cases, the reducing agent and capping agent may be different. Here, citrate acts as both reducing agent as well as capping agent. Modified synthetic procedures of Characterization method are also available. Modified synthetic procedures uses or utilizes reducing agents like NaBH4, that is sodium borohydride, ascorbic acid, simply our vitamin C, as well as different capping agents, tetraoctyl ammonium chloride and different polymers. In a very popular method, HAUCl4 is converted to gold nanoparticles by using ascorbic acid, that's vitamin C, as reducing agent and citrate as the capping agent, and the metal nanoparticles are produced as per this reaction. HAuCl4 gives citrate capped AU nanoparticles in the presence of sodium citrate and ascorbic acid. Here citrate is the capping agent and ascorbic acid act as the reducing agent. Another method for the synthesis of gold nanoparticle is brushed chiffrin method. It is used to produce gold nanoparticles in inorganic liquids that are normally not miscible with the water. It involves the reaction of chlororic acid solution with the tetraoctyl ammonium bromide solution in toluene and sodium borohydride. In this reaction, NaBH4 is the reducing agent and TOAB is both the phase transfer catalyst and stabilizing agent. By this method, we get nanoparticles with the size range 5 to 6 nanometer. Optical and electronic properties of gold nanoparticles. Interaction of gold nanoparticles with the light is strongly dictated by their environment, size and physical dimensions. Oscillating electric fields of a light ray propagating near a nanoparticle interact with the free electrons, causing a concerted oscillation of electron charge that is in resonance with the frequency of light. These collective oscillations are known as surface plasmon resonance, which is an important phenomenon of metal nanoparticle. Localized surface plasmons, LSP. LSP is the result of confinement of a surface plasmon in a nanoparticle of size comparable to the wavelength of light used to excite the plasmon. When a small spherical nanoparticle is irradiated by light, the oscillating electric field causes the conduction of electrons to oscillate coherently with the light. Light incident on a metal nanoparticle causes the conduction band and electrons to oscillate. This is localized surface plasmon. Now, the phenomenon of surface plasmon resonance. The SPR can be tuned by varying the size or shape of the nanoparticle. That is, the size or shape changes, there will be a change in associated surface plasmon resonance. It should be noted that for small nanoparticles in the range of 30 nanometer, the SPR causes an absorption in the blue-green portion, that is nearly 450 nanometer, while red light nearly 700 nanometer is reflected, yielding a rich red color. As the particle size increases, the wavelength of surface plasmon resonance absorption shift to longer wavelength, yielding a solution with pale blue or purple color. That is, color can be tuned by changing their size or shape. Characterization and study of localized surface plasmon. Some techniques frequently used to characterize surface plasmon are dark field microscopy, DF microscopy, UV visible NIR spectroscopy, and surface enhanced Raman scattering, SCRS. The UV visible spectrum of nanoparticles with different size is shown in this figure. It is clear that as the size of the nanoparticle changes, there do a change in their absorption bands. Now, applications of gold nanoparticles. Gold nanoparticles exclusively used in different areas like biology, physics, different areas of science. In biological field, it is highly useful for drug delivery, it is used as sensors, it is used as bioimaging tools. Gold nanoparticle can have a catalytic application. In biology again, it is used for gene delivery. Gold nanoparticles have antimicrobial properties. Tumor or cancer cell imaging is another possible application of gold nanoparticle. Bioassay and biosensor. These all the important applications of gold nanoparticles in the field of medicine and biology. Gold nanoparticles have enormous application as I noted in the previous slide. Sensors. Gold nanoparticles are used in variety of sensors. For example, a colorimetric sensor based on gold nanoparticle can identify if foods are suitable for consumption. That is, we can find out, we can check the quality of a food item using sensors based on gold nanoparticles. Therapeutic agent delivery or simply the drug delivery. Therapeutic agents can also be coated onto the surface of the gold nanoparticle. The large surface area to volume ratio of gold nanoparticle enables their surface to be coated with other molecules. Photodynamic therapy. Near infrared absorbing gold nanoparticles produce heat when excited by lighted wavelength from 700 to 800 nanometer. This enables these nanoparticles to eradicate targeted tumors. PDT or photodynamic therapy is a tumor therapy technique. Diagnostics. Gold nanoparticles are used to detect biomarkers in the diagnosis of heart disease, cancers and infectious agents. Catalysis. Gold nanoparticles are used as catalysts in a number of chemical reactions. Electronics. Gold nanoparticles are designed for use as conductors from printable in situ electronic chips in surface enhanced Raman spectroscopy. In SERS, gold nanoparticles are used as substrate to enable the measurement of vibrational energies of chemical bond in SERS. Now, we are going to silver nanoparticles. Silver nanoparticles or AGNBs is become increasingly important material in many technologies. Silver nanoparticles exhibit highest efficiency of plasmon excitation. Silver nanoparticle is the only material whose plasmon resonance can be tuned to any wavelength in the visible spectrum. Regarding the synthesis of silver nanoparticles, there are traditional methods and non-traditional methods. The traditional method include reduction of a aqueous solution of the silver salt. Non-traditional method includes microemulsion technique, high temperature reduction in porous solid matrices, vapor phase condensation of a metal target onto solid support, laser ablation procedure, gamma irradiation procedure, photo reduction of Ag ions. Challenges associated with the synthesis of silver nanoparticles. There are some challenges during the synthesis of silver nanoparticle by chemical method or non-traditional method. With the traditional method, the major problem is flexibility in the size of the particles. That is, particle size may vary depending on the condition. The major problems for the non-traditional method are often a wide size distribution, lack of particle crystallinity and the cost incurred during the synthesis. Synthesizing a uniform and stable silver nanoparticles with a controllable size is a difficult task. The optimum synthetic method should address all the above challenges. Optical properties of silver nanoparticles. Silver nanoparticles show remarkable physical and chemical properties which are size and shape dependent. That is, their physical as well as chemical properties depends on size as well as shape. The main factors that control the optical properties of silver nanoparticles are nanoparticle morphology, size and shape, the refractive index of the surrounding medium, the medium in which some reactions are carrying out, adsorption phenomena at the solid solution interface, the distance between the particles. Influence of particle morphology on surface plasmon resonance. Silver is associated with the phenomenon of SPR, and some features will control that SPR also. In the case of a sphere, a single peak in the optical absorption spectra was found for spherical silver nanoparticle. For 20 and 100 nanometer sized nanospheres, SPR peaks are at about 370 and 600 nanometer respectively. That is, for a sphere with 20 nanometer size, the peak is at 370 nanometer, and for a particle with 100 nanometer size, the peak is at 600 nanometer. The resonance vanish as the symmetry of the nanoparticle is larger. Influence of surrounding medium on surface plasmon resonance. The location of SPR is sensitive to the dielectric environment or depends on dielectric constant of the medium. As the refraction index increases, the SPAB is shifted to longer wavelength. Resonant Rayleigh scattering spectra of silver nanoparticle in various dielectric environments like nitrogen, methanol, 1-propanol, chloroform and benzene shows this dependence, which is clear from this picture. Antibacterial effects of silver nanoparticles. The antibacterial effects of silver nanoparticles have been used to control bacterial growth in a variety of applications. Introduction of silver nanoparticles into bacterial cells can induce high degree of structural and morphological changes which lead to ultimate cell death. The antibacterial effect of silver nanoparticle is mostly due to sustained release of free silver ions from the nanoparticle. Now let us go through the applications of silver nanoparticles. Silver nanoparticles have several applications in different fields of science. First of all, sensoric applications, or silver nanoparticles can be used as sensors. For instance, peptide capped silver nanoparticles are established for colorimetric sensing. The efficiency of silver nanoparticle based fluorescence sensor is very high. Second one, application in the field of biology and medicine. Silver nanoparticles are highly antibacterial material which is used in water purification, wound care, medical devices and drug delivery. Third application is in the field of catalysis. Supported silver nanoparticles are important in the selective oxidation of alcohols, alkanes and alkenes for the synthesis of industrially interesting products including epoxides and aldehydes. Photocatalysis. Silver nanoparticles are used in photocatalytic process of water splitting, degradation of organic pollutants. Application in the field of optics. Silver nanoparticles are used in optoelectronic devices. Metal nanoclusters are another area of this module. Metal nanoclusters or MNCs. Nanoclusters are ultra small nanomaterials with dimension less than 2 nanometer. They are the bridge between metal atoms and the nanoparticles. This point is very important. This is because metal nanoparticles are bigger in size than atoms but less in size compared to nanoparticles. That is why metal nanoclusters are the missing link between atoms and nanoparticles. MNCs exhibit attractive electronic, optical and chemical properties compared to their bulk counterparts. The stability of metal nanoclusters depends on number of atoms in the cluster, valence electron count, and encapsulating scaffolds. Synthesis of metal nanoclusters. Several physical and chemical methods that belongs to the category of top-down and bottom-up approach are used for the synthesis of metal nanoclusters. Several methods employed for the synthesis are shown in this chart. Chemical synthesis include polyol process, citrate reduction, silver mirror reaction, seed-mediated growth process, pyrolysis, electrolysis, light-mediated synthesis, etc. Preparation methods for metal nanostructures, self-assembly procedure, lithographic technique, soft and hard template directed growth, and biological methods are also used for the synthesis of metal nanoclusters. In general, metal nanoclusters in aqueous medium are synthesized in two steps: reduction of metal ions to zero valent state and the stabilization of metal nanoclusters. Without stabilization, metal nanoclusters would strongly interact with each other and agglomeration or aggregation irreversibly occur to form large particles. The following materials can be used for stabilization of metal nanoclusters. They are thiols, that is thioalcohols containing SH group, dendrimers, polymers, DNA molecule, protein, peptides, and inorganic scaffolds. Luminescence is an important phenomena shown by metal nanoclusters. Metal nanoclusters exhibit strong luminescent properties due to several reasons. The main reason is the electronic transition between energy levels in them or electronic transition between highest occupied molecular orbital, simply designated as HOMO, and lowest unoccupied molecular orbital, that is the HOMO-LUMO transition. Surface ligands play major role in the fluorescence of ligand stabilized metal nanoclusters. The metal nanoclusters derived from gold, silver, and copper exhibit very strong fluorescence. Potential applications of metal nanoclusters. Metal nanoclusters also highly useful in different areas of science. The potential applications include light controlled antennas, photocatalysis for the harvesting of the solar energy to convert it into chemical energy, electronic devices, sensors, plasmonic imaging, surface enhanced Raman spectroscopy, surface enhanced fluorescence, and for medical diagnosis. The therapeutic applications of metal nanoclusters. As in the case of metal nanoparticles, metal nanoclusters are also having several therapeutic applications. The therapeutic applications shown in this diagram include imaging guided cancer therapy, gene delivery, vaccine development, antibacterial agent preparation, used as diabetes medicine, drug screening, and these all are the important therapeutic applications of metal nanoclusters. Thus, in the current module, we discussed about metal nanoparticles, their complete properties, and metal nanoclusters. That's all about this presentation. Thank you.